Good afternoon and welcome to the Arm & Hammer webinar series. Today we're excited to have with us Dr. Rod Mackie and Dr. Phil Cardoso, who will be presenting a webinar on titled Clostridium, an important organism that coexists with cattle and its impact on dairy production. A couple of housekeeping things before we get started. One is we will be recording the webinar. The link to the recording will be emailed to all registrants and attendees afterwards and available on the Arm & Hammer YouTube channel. Participants can submit for ARPAS credits if desired. Details will be included in the post-webinar email. During the seminar, if you have any questions, please submit them through the presenter. We will actually they'll come to me and we'll read them at the end of the overall webinar. So we won't take time between Rod and Phil's talk to actually address questions and that. Just send them on and we can process them and get them ready to talk when we're all done. We have scheduled 1:30 to 3 o'clock, so that's the plan for the overall time. Our first speaker is Dr. Rod Mackie. Rod is a professor of microbiology, Department of Animal Science. He's been with the department 32 years, so an amazing history. Rod's research focuses on anaerobic microbiology and fermentation, molecular ecology in gut ecosystems, degradation of plant cell wall polymers and biomass, nitrogen metabolism, anaerobic waste digestion, and antibiotic resistance. So you can see a number of the areas of focus. The reason that we've been collaborating, Rod, in my career probably over the last 15 years. So welcome, Rod, and you're ready to take the lead.
Okay, so thanks very much for the introduction. I think we can head straight into the presentation. So my job today is to really try and present a historical context for what the term Clostridium means. I want to describe advances that have happened in our understanding of what this group of organisms are, and in the end demonstrate to the extent that we know, answer 3 questions: who's there, who's active, what are they doing, and when they're doing it. And so probably the first 2 are probably better populated in terms of publications, and I'd say the last questions are a little bit deficient. So Commensal Clostridia, if you look through Google and you use that search term, you'll probably find only 1 or 2 scientific publications. And so in general, they're gram-positive, rod-shaped bacteria with spores, and in the modern taxonomy, they're in the phylum Firmicutes, and they make up a substantial part of the bacteria in the rumen and the intestinal tract of cattle. And one of my favorite movies from 30 or more years ago is a Clint Eastwood movie, The Good, the Bad, and the Ugly. And I thought that was a good way of representing these various different phenotypic characterizations of Clostridia. And so the type strain is Clostridium butyricum, makes butyrate, and you consider that to be good. You can see in the picture on the left side of your screen there that they are spore formers. The one in the middle is Clostridium difficile, now actually called Clostridioides difficile, and I'll say a little bit about that later. And that's bad, but it depends on its company, and it depends if you've been taking antibiotics or not. They're widely found in gut systems, and they can be considered commensal. And the last group are the ugly, and those are the perfringens. Those are toxigenic Clostridia, and probably one of the better known ones. Botulinum would be another one. And so I think we maybe need to think a little bit more about what commensal means and what a commensal relationship means. And that means that it's beneficial for one of the partners without harming the other. And that's true, provided they're not toxigenic. And so that means that that definition probably needs some revision. So I wanted to start the considerations about taxonomy by going back to a paper that's now 60 years old. And it's a classic paper by one of the most famous rumen ecologists, rumen microbiologists, and that's Robert Hungate. He formulated this idea about what were the major questions that required to be answered in order to understand a microbial ecosystem. And the first one was, what kinds of numbers of organisms are present? So microbiologists like to know what are the numbers of microorganisms. And this is the classic approach that actually is being used by Arm & Hammer in the study that Phil's going to talk about after me. And then in the good old days, what are the activities? This was mainly done from pure culture studies, not mixed culture, total mixed culture type activities, and then use that information gained from fermentation rates to try and reconstruct what the extent of the activities were in a rumen system. And I've used that as the framework to convert that into modern terminology. And so instead of asking kinds of numbers, we ask who's there. And we do that by using DNA-based abundance and diversity of organisms with today next generation sequencing. Who's active? These are what are the activities, and that'd be RNA-based or based on a functional gene. And the third question is answered by asking the question of function, what are they doing and when? And we do that by at the pure culture or simple mixed culture level by genome expression, transcriptome type analyses. But in the metagenome context, that's for whole rumen content, that would be the metagenome, metatranscriptome, metaproteome, and metabolomics. And so that's how the context for understanding an ecological system has changed over the past 40, 50 years. And so what I did was go back to my earlier sort of introduction to the Clostridia, and that would have been entirely cultivation based. It's all about phenotypic identification and classification. It's taken from Berge's Manual of Systematic Bacteriology. That happens to be the 1984 edition. There have been many editions since then, although the actual content hasn't changed a whole lot, except that there's been a lot of subdivision of many groups of organisms and new species introductions. And so I'm not going to spend a long time going through all of the characteristics. We've mentioned a few of them already, but I just wanted to say that they are differentiated from bacillus by the fact that bacillus are aerobic or facultative, and that the Clostridium is an obligate anaerobe. So importantly, they can be saccharolytic, proteolytic, neither or both. In other words, there's certain Clostridia that actually are chemoautotrophs and use gases, and they're very useful in industrial fermentations. They're very diverse in terms of nutritional growth requirements, metabolism, metabolic pathways, and that's what makes them really an interesting group to study. I think one of the reasons why they're sort of successful in colonizing the gut but able to survive in the environment, in soil and in feeds, is because they're a little bit soft compared to things like methanogens, soft in their oxygen relationships, but they're able to reduce the medium, establish suitable growth conditions, and then proliferate. So if they get in the right place, they can grow very well, and they grow very rapidly. Importantly, they are ubiquitous. They're found in soil, animal plant products, intestinal tract of man and animals. So you can expect to find them everywhere, and it's not surprising that we have them in feed. It's not surprising that we have them in the gut. Since then, since those early descriptions of taxonomy based on phenotype and just fermentation, attempts have been made to try and unify the considerable diversity within the genus. And so the introduction of ribosomal RNA taxonomy sort of really revolutionized classification taxonomy. And Collins in Reading in England proposed a revision of the genus Clostridium based on full-length 16S sequences. At the time, there weren't that many available. Of course, there are many more, although most of them are now short reads. And he proposed to have clusters 1 to 19. And if you go and look at that paper, you'll see that they populated with the species that were available at the time. But the important cluster was Clostridium cluster 1, which was the largest group, and also the one that contained Clostridium butyricum, which is the type species for the genus. So it's the good guy. Since then, there have been numerous other attempts. So in 10 years later, we have that conundrum where there's certain gram-negative staining bacteria. This includes the Selenomonas megasphaera sporomucid group. And based on 16S sequence, they're still included within the clostridial evolutionary lineage, although they can be distinguished by their fermentation pathways. But there are other groups of bacteria that also stain gram-negative, but are phylogenetically, from the 16S point of view, gram-positive. And so for rumen organisms, a very good example would be the Butyrivibrios, another heterogeneous, diverse group of organisms. And then in 2016, Paul Lawson and Rainey, they decided to restrict the genus Clostridium to those that were phylogenetically and phenotypically within Clostridium cluster 1. That includes the Clostridium butyricum. And so now you see that there's this big proliferation of other groups that are no longer called Clostridium, but before they were linked based on simple phenotype. So as we go through the rest of the talk here, just think about what we mean by classification rank and the nomenclature that we use to talk about that. And so the one right at the top, that would be the species level, and going up or going down the pyramid, genus, family. And so for short reads, many of the classifications only read to family level, the Clostridiaceae, or even just order, the Clostridiales. And it's very common now to just talk about a phylum, the Firmicutes, and people refer to those 3 major phyla in gut systems. But I wanted to just pause for a moment and emphasize the bottom 2 points here, and that is that that there's enormous strain variation within each of these species, and the strain variation is so large that really the type species description doesn't fit many of the existing organisms that are now classified with it, and importantly, that strain differentiation is not covered or captured with 16S sequencing, and it's certainly not captured with short read technologies. So just bear that in mind. So there is a lot of diversity and there's a lot of variation, and in gut systems, that variation is often at the strain level, less so than at species level. It's part of the structure of gut systems. So fine branching at the end of these lineages. Okay, so the next era was one which I think was really quite positive. And so we were able to differentiate low GC gram positives from high GC gram positives. And it was a bridging type of technology. It was a bridge from the original taxonomy that included the percentage G plus C in a genome, not in a sequence-based way, but just in terms of a melting profile. That's very useful in describing community composition, the structure of bacterial and also archaeal populations, pathogen populations. Okay, and so within the low GC gram positives, and that would be around 22 to 55% for the Clostridia, it includes Clostridium, but it also includes Bacillus, Lactobacillus, Streptococcus, Ruminococcus, and a bunch of others. It's quite often that you see another group of sequences now in modern phylogeny that relate to the actinobacteria, and the most important representative there would be bifidobacterium. So in young animals, breastfed babies, of course, you're going to see actinobacteria, and those are represented by bifidobacterium. So we don't have a very nice arrangement like this for rumen systems, and so I've just used this one from the human colon, and it shows the two major clusters, clostridial clusters, that are no longer considered to be Clostridia, but many of them still have the name, Clostridium clusters 4 and 14A. And you can see on the left-hand side of the screen, Clostridium 14A, a big one. It's also known as the Clostridium coccoides group, and I've boxed that in red. And below that, you can see that there's a bunch of others that are now classified as something else, but they are non-spore forming. And those include things like Peptostreptococcus roseburia in the human gut, Ruminococcus, Torquis, Canavis, and in the rumen, albus and others. And in the cluster 4, that's a much smaller group, but includes the Clostridium lepton group. And there's another interesting group in this particular publication was interested in bile acid production. And so those are groups of those are type strains or strains of bile acid metabolizing Clostridia. And so they're probably important in some of those transformations. And then the ugly guys, those are the ones that are in the bottom box on the right there, botulinum, cadaverous, difficile, perfringens that are found in animal guts. So right, so now we transition to the present, what's going on right now. We're in the age of metagenomics and metatranscriptomics. And so now we use the concept of phylum level taxonomy to describe microbial communities, and shown in the pie diagram on the left is a representation of what's found in the rumen, and it shows in blue one of the major phyla, that's the Bacteroidetes. It shows in red the firmicutes, and in green the proteobacteria. But these fractions, pieces of the pie, vary a lot depending on diet, depending on age of animal, and you can see from the box and whisker plots on the bottom, there's still quite a lot of variation within the Bacteroidetes and the firmicutes. And so we're interested in that red fraction of the pie that includes the Clostridia. So I scoured the literature looking for more data on Clostridium, and it's not easy to find, but there are a couple of studies that use this modern next generation sequencing to look at populations of at least firmicutes, and this particular one describes the effect of age. And so if you look on the left, that bar graph, that would be straight after birth, 1 to 3 days. The next one is 2 months of age. The next one, 6 months of age, and then 2 years of age. And this particular study does show that the Bacteroides are more predominant than the firmicutes, but this is a dairy cow study, and that's done in Israel under their conditions. Okay, so now let's have a look at where these groups of bacteria are located. And across the bottom, you can see that there's metaproteomic data, so a level of expression. And to the right, the 16S RNA gene levels. And let's just focus on the right set of columns there. And for this particular study done in Germany, you can see that everything that's in green or a shade of green are firmicutes. Everything that's in blue or a shade of blue is a Bacteroides or Bacteroidetes type phylum. And remember that The fibrobacters, which are very low on the metaproteome side, are the purple bars, and these also used to be Bacteroides succinogenes, but now they have their own genus based on certain criteria. You can see in this particular study also that Bacteroides outnumbered as a relative abundance the firmicutes. So, well, what are they doing? And so when you do things like metabolomics and when you do things like proteomics, you're able through using searches to databases, find out what's the average abundance of genes that are involved in certain fermentation pathways. And I've just pulled out one of the tables here. And what it shows is that in terms of butyrate production on the left, formate production on the right, that the firmicutes are the major players in that production. In contrast, the Bacteroides are really important in terms of making propionate and acetate. And of course, remember that the Bacteroides, they're defined by making succinate. And succinate is decarboxylated to propionate. And so that's the reason why propionate's assigned to the Bacteroides, or major portion of them are. Although firmicutes are involved in both propionate and acetate production. So you can see some green up on the top of the propionate and acetate set. Okay, and then once you've got all of that data, and this particular study tried to present a schematic representation that combined their metaproteomic, their 16S sequencing, and metabolomic analyses to show which were the most abundant bacteria involved in these various activities. And there are three diets on the left: be corn, the more yellow; hay in the middle; grass on the right, that's green. Although it's hard to distinguish which are more active on what. But I think what you can see is that the Prevotellers in the rumen are really, really important in many of the activities. And unfortunately, the Clostridia are not major players in terms of abundance in this particular study. And that reflects the previous two slides that I've shown you with data from that paper. Okay, so another study that comes from Nanjing Agricultural University looked at populations along the intestinal tract of cattle. It's a decent introductory study, but why don't we just focus on the mucosa? So that would be the bar charts on the bottom there, and focus on the mucosa. And you can see that the firmicutes are the blue bars right at the bottom. So digest on the left, mucosa on the right, and starting on the left, it runs from rumen, reticulum, omasum, abomasum, duodenum, jejunum, ileum, cecum, colon, and rectum. And so when you go across to the box and whisker plots, and just let's look at the top box here. And so that would be the firmicutes. And so initially they kind of low. Once you get to the jejunum, ileum, duodenum, jejunum, and ileum, the numbers are much higher, and Bacteroides are much lower. And so this indicates that maybe a lot of these activities for Clostridia in terms of being commensal bacteria are actually post-ruminal. And you can see that Bacteroides are initially higher, and then they decrease a lot and increase as you go down and get to cecum, colon, and rectum. So I just wanted to add that actually we have a very nice study now based on the study that we're doing with Urbana-Champaign that Phil's going to talk about. And so we actually sacrificed 16 of the 64 cows that were in that study, and the analyses of that work are ongoing. And I think that's going to be really, really important in terms of our understanding of where these organisms are and what would be their absolute abundance. Okay, so what do they do? And so, hang on. before I do that, let's just drill down a little bit in terms of now what are the family level affiliations. And you can see that once you do that, you tend to lose a lot of the firmicutes, and so the clostridiales, that sort of brick red type color, tends to be present, but not in the lower gut. So there's other organisms that are taking up that taxonomic space, and you can look down at the bottom and see for the mucosa that there's amongst the most predominants, the Clostridium are not in there. Okay, so then what are they doing? So I've just prepared 2 slides, and I don't want to spend a lot of time going through them, but Clostridium, they ferment a wide variety of carbohydrates. They're very good at degrading insoluble plant cell wall polysaccharides. The model for the cellulosomal concept is Clostridium thermocellum. That's a thermophile, but the best model for mesophile is cellulolyticum. So those are non-ruminal organisms, but they found a soil degrading plant material. They're good at cellulose degradation. They ferment soluble sugars and make mainly burate and formate. Also, they make a lot of hydrogen, and so that's important in terms of relationship with others. Protein, they're probably better known for that, protein or nitrogen metabolism. It varies quite a lot. Some are very high in deaminase activity, others aren't. They're known for this Stickland reaction, and Jim Russell was quite successful in showing that these organisms were present in the rumen, not in large numbers, and they are hyperammonia producers. They also decarboxylate amino acids, making potent things like histamine, active compounds. Also been found in mimosine degradation. They're important in other transformations such as biohydrogenation of long-chain fatty acids, and in monogastric type animals, bile acid metabolism. So what we want to do in microbial ecology and diversity is look at things that can be overcome without a cultivation base. But I want to emphasize to everyone who's at the webinar that in order to present a decent paper and a complete piece of work, you need to measure real abundance, concentration or level. I said microbiologists like numbers, and we like to know a concentration. Numbers of organisms per gram or per mole. Diversity, and that's what virtually all of the publications today are. They're just measures of relative abundance, not absolute abundance. And they do give you an index of alpha and beta diversity, but that's not true abundance. Most importantly, you need to measure function. And function is not sufficient just by looking at a DNA metagenome. You need to measure some real functional attribute. And for the rumen, that could just be something like VFA production. So who's active? Those are questions of function. What we want to know is, just because a gene's there, does it actually mean that it's being expressed? And so to do that, we need to make RNA, and we can do that. But remember that in all of these cultivation independent approaches, we got around one problem, but we've introduced a bunch of other problems. And so there's a whole lot of other biases that go with all of these approaches that start not only with sampling, and that also applies to cultivation-based approaches, but things that relate to DNA extraction, PCR bias and amplification, additional bias when you're using RT-PCR. All of these things are very important, and so they need validation. Another way of doing it is to focus on a functional gene, and I think probably the best known examples of those are probably the MCRA gene for methanogens, so not Clostridia, or things like the DSR A and B genes for sulfate reducers. And you can do the same things with the functional gene that you can do with the 16S gene. So you can make sequence libraries and use modern next generation sequencing to find out what's happening with functional genes. Probably one addition I could make is the NIF-H gene. That's the nitrogenase gene. So for something like nitrogen fixation, which some Clostridia also do. So when are they doing it is an important question, but it's a question of time and experimental design. So what you need to do is have a sound initial hypothesis, an appropriate design, experimental techniques to answer the questions, and use a time series. So an example would be a number of samples collected before imposing an experimental treatment, a diet or something, or an additive, during adaptation, after stabilization, and after removal. And you need multiple samples at all of those times to get a time series that allow you to make a proper pattern of what's actually happening. So you don't want a snapshot, you want some sort of decent time series. And so that's what you get in a transcriptional profiling type approach. And really those are the gene expression. So that's from messenger RNA, can be from proteomics, can be from metabolomics. And what you want to do is identify the intrinsic patterns and relationships for all those either genes, proteins, or metabolites. And the final objective then is to provide some sort of higher level functional analysis that resolves structural and functional maps of any system you're working in, but in our case, the rumen, and these are few and far between. So the red message I have across the bottom is really important. Say, look for causal relationships, not statistical correlations or associations. So most of the studies are just the beginning, and they're not the end of the study. They need proof and validation. Okay, so the two players that we're really interested in the studies that Phil's are going to describe are the Clostridium bayerunckii, and the other one would be Clostridium bifermentans, now referred to as para-Clostridium bifermentans. So it's a little bit outside because it's in cluster 11. It's not strictly Clostridium cluster 1, like the bayerunckii on the left. So they're both mesophiles. And fermentum, it's known by its name for fermenting both carbohydrates, and that's, but not starch, and amino acids. And so that's why it's called bifermentants. Bayerunckii, named after a famous Dutch microbiologist, Bayerunk, is also interesting. But its interesting features that it's sulfonogenic. So it converts glucose first into acetate and butyrate, and then into solvents, butanol, acetone, or even isopropanol. And again, to emphasize the point, they found in soil and intestinal tracts, both of these organisms. So it's not surprising that they're well represented, and they're a decent group to study for trying to modify or manage rumen fermentation. So my last bit of the presentation is just a short presentation on the solvenogenic Clostridia, and the ones that are of interest to us are in the top group. So it's acetobutylicum and bayerunckii, both in Clostridium cluster 1. They ferment starch and sugars, and they make those end products. And they do that in a very interesting cycle. And so part of the cycle that's shown in green on the top is an acetogenic phase when vegetative cells are growing rapidly, and the pathways that result in formation of acetate, butyrate, and even a little bit of lactate are shown on the left in green. And they sporulation is triggered, so they sporulate, and during that process they start to reassimilate acetate and butyrate, shown in blue, and make from acetone. From acetate they make acetone. From lactate they make ethanol. From ethanol they make lactate. Sorry, wrong way around. Lactate they make ethanol, so it's the ABE fermentation. Acetone, butanol, and ethanol. And on the bottom, that would be reassimilation of butyrate to make butanol. And so it goes through a cycle like that. And sporulation is triggered by low pH, and that's shown in this following slide here. And so the acetogenic phase shown on the left, this is for acetobutylicum. Bayerunckii would be almost identical to this. And the top graph shows the effect of what happens to pH. So a rapid drop in pH to round about below 5, round about 4.7, 4.8. Rapid production of first butyrate, the top loop on the bottom, then acetate, and then reassimilation of those acids to make butanol, the biggest end product, then acetone, then ethanol, and declining levels of acetate and butyrate. And so that's the classic ABE fermentation, and that would be carried out by something like Bayerinkii. All right, so to conclude, I'd just like to sort of combine a lot of my the talk and my observations and analysis of what's going on about molecular microbial ecology and gut systems. And this is not specific to the rumen. I think we'd have to knock off a few of the pluses for the rumen, because now we're no longer in the lead. We're no longer in the vanguard with these systems. We're following other environmental microbiologists. But I think we've done a pretty good job with diversity, and I've marked that as 5+. Ecology, less, and that's because people aren't making the proper measurements, and people are just tending to use relative abundance to do that. And so for the rumen, I'd say we're there only about a 3. Some other environmental systems, maybe we're a 4. And so on the left, not bad. But we need to be really critical about what's happening to the right side of this picture. So structure function, really deficient, especially in the rumen. Those studies are hard to do, and we don't know a lot about those. And in terms of evolution, this is really just the beginning. So in the future, we're going to be looking towards trying to add to our knowledge on the right-hand side of this equation. Okay, and so my final slide is just to say that it's not sufficient to do cultivation-independent studies alone. What we need to do is use cultivation-dependent approaches combined and amplified with cultivation-independent approaches, and use that to advance our understanding of this amazing group of organisms. And again, just remember that what you need to do is think about true abundance, diversity, relative abundance, and function. That's where we need to get. And so with that, I'll close my talk and hand over to Phil, which I'm in the process of doing right now.
Okay, and again, remember to submit your questions. As Phil gets his slides set up, just want to introduce Phil. Dr. Phil Cardoso is an associate professor in Department of Animal Sciences, University of Illinois. He's been in that professor position for the last 8 years. Phil's research focuses really on addresses the most important challenges faced by the dairy industry through understanding nutrition and reproduction. He engages dairy producers to implement data-driven best management practices into their operation. He studies mechanisms of metabolic adaptation from gestation to lactation and the impact of nutrition on metabolism, reproduction, and health. And today, Phil's talk is titled, The Challenges of Non-Toxigenic Clostridia for Dairy Cattle. And with that, Phil.
Thank you very much. I would like to thank Tom for the introduction, for the invitation to this webinar. Also, Elliot, that is watching us, that is coordinating all this series. And I think it's a, through this tough time, I really appreciate you guys putting this together to try to bring some of this knowledge and hopefully a little bit more digested, since you have this chance to present to you guys and answer some questions as well. So as you can see, I have a little bit of a southern accent, and that comes from Brazil. That's my home country. That's a map there, and actually what I'm showing you there is some very interesting places where we have wine in South America. So I'm showing you the state of Rio Grande do Sul in Brazil, the southernmost state. And also, I'm originally from, let's see, I'll figure this out. From this city here, Porto Alegre, that's the capital of this state. And as you can see, all these numbers, they represent places where you have wineries. So there is something special about those places that make grapes great for this type of product. So here is one of those wineries. Actually, it's a region, 1.5 hours from Porto Alegre. You can drive and you can visit a series of wineries, very famous, and they have pretty good wine. But you can see that the map already doesn't show you. It's a 2D map, right? It's not showing you the whole curvature of the terrain and how the micro climate can change within one winery to another winery. Here you can see just one winery. You can imagine already how different those plants, those grape plants can be, right? And that's one of the things that they do. Actually, they created seal that goes in every bottle, and that means that at least 80% of the grapes in that bottle came from this particular region, right? And it's that means that it comes from this winery valley. That's how they called it, the little translation for this seal here. And that not only is a certification type of recognition, but also it's telling us that everything particular to that region is going to be represented in that wine. And throughout the grapes and the fermentation process, you'll be able to feel it, right? Or taste it in your mouth. And they say that's the best region for Merlot in the whole world. Who knows, right? I've drank a little bit of those and I really like it, but I'm not a really wine connoisseur. And I'm also sure that I could do this type of exercise in any country. I could do the Napa Valley. I could go to Rod's place in South Africa, and I'm pretty sure I would have some regions and some wineries, and they would have their own little thing. But these guys here, they're trying to commercialize that by saying, hey, you get this symbol here in your bottle, 80% of the grapes are coming from this particular region here. And that comes to what we call that particular things and all that characteristic, we are calling the microbial terroir or wine terroir. And it's just this effect of the bacteria, the yeast, and all other microorganisms that may be in that vineyard, right? And Rod was talking about a lot of ways that we can identify now different species or genera of bacteria and other microorganisms. And actually, they went through trying to understand not only the pathogens that are very well known for a long time that can cause diseases in those plants, but also some of the commensal or some of those microorganisms that are present in different parts of the plant or even in the soil. And that pretty much represents that whole microbial terroir. And that's very particular, and there are research talking about each winery or which region may have, and even within a same winery, depending on the size, you may have different types of terroir or microbial terroir, right? I'm sure that Tom will talk more about this concept just after me. But one of the questions is, how about this, right? So after I graduated from vet school back in Brazil, I worked in practice for 5 years before doing my master's and PhD, and that's one of the dairies down there. And you can see, how about that microbial? Those cows, you can see they're on pasture. They are maybe a little bit more dirty than I expect. How about what is that microbial terroir? Can we define that and what it's out there right now? So it's a very, I would say, for cattle would be a new concept, and I think now we have the tools to identify those guys. But in a dairy farm system, I would say that everything that brings those farms or that farm similar to each other, right? And there is research talking about that all those microorganisms, they may impact not just the diet of the cow or the health of the cow, but also, as you saw, for example, those cows that I showed before, they were a little bit dirty. So that can come to the milking parlor. That can be a potential hazard for mastitis and other diseases, or even to show up in the milk for consumers, right? And more recently, and that's where I think Tom was going to talk a little bit more about the survey, that they really nail down what are some of this bacteria that are happening in those dairy farms in the US. And so they collect samples, TMR, corn silage, haylage, all over the United States. And this is how the Clostridium kind of shook up, right? So bigger majority, what they found was Clostridium perfringens, and I'm gonna divide them here in toxigenic. That would be this perfringens where they have toxins, right? Where you guys probably know some of the diseases. There is one that is very common, or was very common here in the Midwest, where because of the production of toxins, now they can cause this necrotic enteritis. And we see in all species, but you can relate in dairy cattle with the HBS, or hemorrhagic bowel syndrome. That's not unique to the Midwest. That's not what I'm saying. Other places in the world, they have the same thing, right? But however, all these non-toxigenic type of bacteria, that's somehow less understood. What are their role? We know from this research that yes, they are out there, and cows have been eating them. They are in a, more than 25% of that material is contaminated, or it has this type of bacteria, this Clostridium non-toxigenic. So what do you know about them? And that's kind of one of the things that we want to understand more. And that's why we had to put up a team to understand that. And my grad student here in the middle, Fabiana, she did an awesome work. So I have to acknowledge her on all the work collecting the samples and putting up some of these results. And also she was able to build a team around her to collect samples and everything else. You guys are going to get a feeling for how taxing this type of work was on her. I just realized that she's wearing the wrong hat on that picture, but that's fine. I'll have to fix that later. So what was our hypothesis was that, hey, we're going to give this non-toxigenic Clostridium mainly by fermentants, and this is going to be bad for the cow. That's going to hammer the cow or the production system. Something bad's going to happen. And the objective was, okay, we're going to feed this as an oral challenge, and we are going to evaluate the health and metabolism. So this is somehow of a pilot, or trying to understand this. So it's kind of a pilot study to see where we go next, right? So for that, we had 41 cows, right? So we fed, or we challenged those cows, like I'm going to show you in a little bit, for 70 days. We have 1 week of adaptation, and then 9 weeks of feeding the Clostridium. They were all in tie stall, like you can see in the picture. One thing that is important to highlight, those cows were past peak, so pretty much mid-lactation cows that we fed them all over. Like Dr. Mackie said, a section of those cows, they were sacrificed to understand a little bit more what's going on inside. I'm not going to show that in here. That's going to come from Dr. Mackie and Tom later on. I'm going to show you the first 9 weeks of that trial, the production section pretty much. They were all multiparous, and that's some characterization. So how did we divide this treatment? So we had 4 treatments. One control, we had no addition of this non-toxigenic Clostridia by fermentants, okay? And then we had 3 other treatments, low, medium, and high, according to how much Clostridium they got, based on CFU by gram of that TMR or that inoculum, right? So how did we figure out this low, medium, and high? So that's the TMR. We were collecting TMR from each one of those cows. They were all in tie stall, so we would collect, group them by treatment, right? And we would send that to Arm & Hammer, the Arm & Hammer team in Waukesha, and then they would measure how much Clostridium would have in that TMR. So here, for example, all over the trial, you can see what was the count of total Clostridia in the TMR, okay? So we plan to do that every 2 weeks. So we have a sample on the week 1. Week 3, we see that spike actually was very coincidental with some hay that was being fed that had a lot of mud and a lot of contamination. We said, hey, how about we just get a better hay? And then everything went down from now on. And then we decided, hey, let's collect every week. So that's why from week 3 on, we have weekly samples on Clostridia. So what we are doing is, according to how much Clostridia was in that TMR, we would, the guys at Arm & Hammer, they would grow that Clostridium to 1 log higher from what we had, put in layer vials, sent to us, and then we would feed that the following week. Okay, so this was what we found in the TMR, and what we were doing was this. So we received that during early week. We would receive the inoculum, and then we would feed that to cows. And then we'd feed in 2 ways. In the morning, we'd put that spiked in corn inside gelatin capsules, and then we would give it to the bolus. I say we, but you can see Fabiana there, not smiling that much anymore. She lost the hat. But we would do cow by cow in the morning, that gelatin boluses with half of the dose, daily dose, and the other half of the daily dose would come as a top dress, and would of course make sure cows were eating everything. So that's how we determine that some cows, they got a high dose, a medium dose, or a low dose all over the period of 9 weeks, okay? And here's the diet. I think that's something that we need to really pay attention and kind of start discussing a little bit more, is the ingredients of the diet. So we have a, you know, we fulfill the requirements. We are not wanting to do anything fancy, but we are using rumen protected amino acids like we do. We are using some blood meal that maybe some places that would not be allowed. So it's just a 40% corn silage diet, very typical for mid-lactation cows here in the Midwest. And crude protein, you're not going to see that 17, 18% because we are doing the rumen protected amino acids. A pretty good fiber component. And if you think that 25% starch is in the low side, 30% in the high, we were right in the middle there, right? So we're not a super high starch diet that you may see in fresh cows. But remember, we are mid-lactation cows. So we were not super hot, but we are not super easy as well. Like you could see maybe in a California diet, what we see much lower starch at 23, 24%, right? And here, what I'm showing you, okay, the same way that we would be sending samples of TMR to the lab to count for how much Clostridium would have, we also send fecal samples. And here, we missed week 6. That's why you don't see results for week 6. But you can see that control, low, medium, and high, and I took it out the standard deviation here because that would make everything the same and a messy, right? But you can see, for example, that it relates a little bit, especially with as time goes by, what cows got in the TMR. However, if you remember, first, I think to highlight the dimensions, right? How many CFUs per gram you have in TMR and fecal, they are very different, the amount that you can find. So even though we had that spike of, we could say, natural Clostridium on week 3, that was not totally represented in the fecal samples, at least not for control, low or medium, or even high, right? Imagine that all the cows got that spiked per se natural Clostridium, but I think there's the treatments they were getting that was getting to the cow that we saw was coming out in the fecal samples as well, right? And so some results here, we saw that the cows, now we kind of got a little bit a tricky part, right? Because now the cows that have a higher concentration of that non-toxigenic Clostridium, they tended to eat more than the cows that received a lower concentration of that Clostridium. Milk yield, we didn't see any statistical difference, right? And remember that, again, mid-lactation to end lactation, that for you to move intakes or even milk yield is much harder than if you do that in the beginning of the lactation or even throughout the transition period, right? And very interesting enough, this is research is fresh out of the oven. This is was presented at the ADSA this year online. And what they did was in a dairy farm, commercial farm. So I don't have information exactly how they measured the intake and how they really nail down if people were feeding or not, how they were feeding, if they checked if they were eating everything. I have no idea, but they had 2 treatments One was a DFM that had one of those non-toxigenic Clostridia, the Clostridium barranquii, and yeast gets Picha, all are in the rumen naturally. But what they saw was when they fed that pretty good number of cows, 140, dairy down there in Argentina, not here in the US, they saw that starting at especially week 9, you can see that there was a higher milk production or milk yield for those cows receiving the DFM, right? So here now we start changing a little bit from our hypothesis, but we are seeing a little bit more intake with the high group that received more Clostridium non-toxigenic. This other work here is showing when they gave that DFM, combining those two, they were able to see a greater increase in milk, and they quantified that to 2.8 kilos, so roughly 5-6 pounds more in that whole period. But look, they went for 31 weeks on that trial, right? So already gives us an idea that maybe we did that for 9 weeks. Maybe should we be doing for more time, or we would see more effect, or if we had it fed to cows early in lactation or even during the transition period. And I'm gonna get why, but of course we had to do the pilot to understand what's going on, right? So other research out there, they probably didn't measure intake, but they saw difference in milk. And even though the diet is kind of similar, I mean, it's a poster, so they don't describe really well, but I can already tell you that corn silage in Argentina is very different than corn silage in the US, where fiber digestibility would play a big role when you're feeding TMRs down there in South America versus the US, and corn as well, when you have a dent corn versus a flint corn down there. Now, milk components, we didn't see any difference. Again, we are feeding mid-lactating cows the right amount. They're producing good fat, good protein, no difference there. Now we start seeing a little bit of, and again, this is a pilot. We have 10 cows per treatment, so we cannot be very audacious here or setting down the last true and last word on everything. But that seems to be that milk urea nitrogen was lower for all these cows that we were feeding the Clostridium than the control cows. And I think I saw one of the questions before Rod was done asking about, hey, these bugs, they are digesting, or they are using, or they may be releasing some of these peptides or amino acids. How about down there, down in the gut? Is that going to make a change? Well, if we hypothesize that the cow is getting more amino acids through increased digestion by this Clostridium or other microorganisms, that could mean that they need to recycle less nitrogen, and that could be showing in this milk urea nitrogen being lower because they are just getting enough, while control, they still need to recycle more nitrogen, and that's why you have it more in the control cows. Maybe a little bit of stretch is a tendency, but it's definitely a way to go with some of these other things that I'm going to show you right now. So rumen pH and fecal pH, we were able to measure. Again, rumen pH, we were measuring all those 40 cows every week, and we were doing that with a probe, right? So we know the saliva contamination and everything else. However, there was a treatment by week interaction for both, and we can see that the control cows, they seem to be having a higher pH than the other cows, especially in the first 3 to 5 weeks, right? So there seems to be some modification or modulation of rumen pH when we are feeding those Clostridium non-toxigenic versus control, right? One opportunity that we had some rumen cannulated cows. So we had roughly 4 per treatment, right? We had 15 cows. And what Fabiana was able to do there is that she can catch the rumen papillae and collect that, and we can do some gene expression work. So we can see what are the genes that are being more or less expressed in those cows in the different treatments. And what we saw is that for this gene, for example, toll-like receptor 2, and specifically, this is a gene in the rumen papillae, remember, that has some kind of role in interacting the tissue with the bacteria, specifically gram-positive bacteria, right? So we can see that seems like control is having more expression or would be relating more to that than any of the other cows that were receiving the Clostridium. So here I start thinking about how is this Clostridium being able to shift the rumen environment in a way that now I'm having less of that contact in the rumen papillae. And again, I don't have the answer. I think the next trial hopefully can, the samples that we have from this, other samples we have from this cows, that we can answer that. We didn't see any differences in the VFA, but again, remember we collect those rumen fluid samples through a probe, right? Now, another genes, and we have a bunch of genes, but I'm showing you the ones that we saw differences, right? Those two clouding proteins here, or enzymes here, they're related to code for proteins involved in the tight junction. So how connected, and you guys heard about that leaky gut type of concept. So tight junctions are the ones that are not allowing actually bacteria to go from the rumen to the blood circulation of the cow, right? So something needs to be tying those cells together, those epithelial cells, in the case of the rumen papillae. And you can see that you have a little bit more expression of that on the cows that received the non-toxigenic Clostridium. What again is telling me, okay, now I had less of that communication, and now here I'm having to have more expression of those tight junctions, perhaps because other types of bacteria are trying to cross those cells, but that can be, again, a little bit of stretch. But there was already research showing that cows with acidosis, they have higher expression of those tight junction genes. So in the case, we showed that there was some manipulation of rumen pH going down, more acidic. Now I have the need to go higher in those tight junction cells like this one. And when I show you this gene also, now not related to tight junction, but related to the metabolism. Remember, the rumen papilla is an active tissue that is trying to metabolize all that VFA that is coming from the rumen microbes. Now you can see that the control has this higher expression for this enzyme that is involved in metabolizing BHB and pretty much more ketone butyrate type of VFA, right? I mean, we didn't see differences, but I don't think our collection method was the gold standard for VFAs anyway. But you can see that perhaps these cows with Clostridium, they are actually targeting a metabolism more related to perhaps propionate, or related to that story of the lower pH and the more amylolytic type of bacteria in that rumen, or just by improving fiber digestion, now I can release more of those sugars present in forage, and then that's kind of how my rumen papilla is adapting to better metabolize. And remember, I'm not sure if I told you, but we collected those rumen papilla by the end, so the cows had the maximum exposure to all those bugs. So in summary here, what I have for you, we had this cow, we collect blood samples. I didn't show you some of the panels. We didn't see a big difference. So there's not any physical or physiological changes in these cows that they're getting sick that you can visually see. However, there was this tendency for this non-toxigenic tending to increase dry matter intake. We saw differences in the rumen papillae. So we truly believe that this bacteria somehow is shifting the microbiome, if not by itself causing some of these changes. And we also saw that tendency for mucourea nitrogen that can be very well explained by how this type of bacteria uses the substrate, like Dr. Mackie was talking before. So if I bring back our hypothesis, right, that these guys are going to be detrimental, to be honest, at this point, I'm not convinced of that. I may have to change the hypothesis or just deny my hypothesis here. Like in Connecticut, we are full of surprises. And I think we need to understand more that I think depending on the system, depending on what's the diet, how much starch you have the diet, how much sugar, what type of cow, you may have these guys being the villains or not being the villain, or even being involved in helping with fiber digestion by changing the rumen for more amylolytic, low, because we saw it lower rumen pH, can improve fiber digestion. That leads to that tendency to increase the intake, and that would explain all the changes that we saw in that rumen papillae. But I think it's going to be somehow different in each diet, and that's what I had for today, Dr. Tom.
Okay, thanks, Phil.
You're
Again,
welcome.
don't forget to submit your questions. The questions are anonymous, so there's no issue there. So just type it into the chat box and it'll go to the organizers. So I'm going to take it from here now that we understand a little bit more about the Clostridia as a genus, some new findings, some new diversity understandings, understanding what we're doing for the future. As Rod said, you know, some additional work needs to really understand more about structure, function, and evolution. So that's really an important goal for where we're all headed. But I do want to share the work that we've done to date, looking at Clostridia populations in U.S. dairy herds, and then thinking about, based on those results, what are the predisposing factors for different levels of Clostridia in the gastrointestinal tract that we measure through the proxy of a fecal sample. And then switch a bit and talk a little bit about solutions and mechanisms to control. So with that in mind, let's get down here. Okay, so Phil alluded to terroir. That's the word we use to really understand that diversity of microorganisms within an animal, and within a system that animal exists in. We can use our microbial terroir platform then to characterize those Clostridia populations, screen them for sensitivities to bacillus, which we actually use as a commercial way to control those Clostridia, formulate specifically to a region, or in very large farms that are 5,000 cows or larger. We can actually do this on a farm level and customize an approach, validate its performance, and then continue that process, because as we know, Clostridial diversity is going to be changing over time based on the feed sources, and we'll kind of talk about that as we get through the slides. So this is the work that Jesse and his team has been doing since about late 2015. This is the colored map of the regional U.S. survey. There's 9 different regions. We'll come back to those. There they are by name and color. We really have 2 parts of the database. One is that we have enumeration results that give us enumerations of total Clostridium, Clostridium perfringens, and then we have the full molecular analysis in which we have characterized genetically the isolates. And what you see here is a combined for each region, the total farm sizes, as well as the individual states in the box above. So as I mentioned, it's 9 regions. Today, there's over 20,510 fecal samples from 509 farms representing 30 states. And if you look at that number, just a second, I'm moving my box, there you go. Samples from farm really represent then about 1.3 million dairy cows of the roughly 9 million dairy cows in the US. So we're well over now 12% of the total dairies that have been sampled. So what I want to walk through is that sample processing and what we do. I'll just call this up to make it a little bit easier. We collect the forage, the TMR, and fecal samples and relevant commodities that may be suspect, and we bring those into the lab and enumerate on a selective media, differential selective media called tryptone sulfide cycloserine. We pick 5 colonies from the highest dilution and then characterize those one of several ways, and actually, well, the first way is to actually find out if those isolates actually produce an alpha toxin associated with Clostridium perfringens. And if those do produce an alpha toxin, we want to know how diverse is that population of the Clostridium perfringens detected within those samples. Samples that do not contain the alpha toxin will actually run a 16S sequence PCR so that we can actually PCR first and then 16S sequence for those that we can't fully identify, so that we get a phylogenetic understanding of what is the diversity. And that's a common pie chart that we'll show you as we go through the data sets. So as I step back now, we're going to actually show you the results of the US herd averages. On the left hand is the calves, on the right hand is the cows. And you can see in terms of Clostridial prevalence, this is total Clostridia. Fecal samples were actually showing that 97.9% of the samples were tested positive for Clostridia. When we looked at Clostridium perfringens prevalence, so now we go down and look at just those samples, fecal samples that contain Clostridium perfringens, we found that 80.4% of the cow samples actually contain. So there's a prevalence within the US herd. So as Rod mentioned and also Phil mentioned, common to the soil, common to a lot of the different environments on the farm. It's in the water, it's in the feed, and we'll show you some of that data as we go through this. What I want to do now is kind of walk you through the variation of the data that we actually see. Again, on the left-hand side will be the calf fecal results. On the right-hand side is the cow fecal results. And just to set this up, what we have here is the total plate count, or the total tryptosulfite cyclosine agar. So that's total Clostridium and CFU per gram. And then these are the various regions. And you can see there's so many dots here that it actually kind of looks like a, just an elongated smear of color. And you can see right away that what we see, in fact, is a very big range of levels of total Clostridium, both in calf and cows. And in fact, for us to, in any region, see 6 to 7 log variation between fecal samples is not unusual. So I'm going to drill down now and look at what that looks like. So this is the number of samples on the left-hand side, scale, and actually Clostridio, so we can actually, again, get a better fix of what we're really seeing. And the mean samples, 51% of the samples fall in what we consider to be a moderate risk, and we'll talk about risk levels. But what we're saying is that we have 51% of the total samples were actually between 100 and 1,000 CFU per gram. When we look at cattle that actually have HBS and cattle that are in digestive disruption or other health problems, we actually see that the populations of Clostridium, be it toxigenic or non-toxigenic, can actually exceed 1,000, which we put as a high risk and shade that red. And this is how we've been able to actually use this data to understand more about where are the risks and how we have to think about the challenges of what we're feeding and what that impact is on overall herd health and performance. So the next level is now to actually look at Clostridium perfringens. Again, you can see how dramatic that data is. It's a 6 to 7 log variation within farms. Each one of the dots actually represents an individual fecal sample. There's a number of farms, and I'm going to break out now in the next slide. Oh, just here's again a histogram showing again the highest frequency actually comes in that middle range of 100 to 1,000. Now representative of only about 36.8% of the total. So we see less Clostridium perfringen risk than we do in total. When we zoom in and look at individual farms, I wanted to kind of show you the data that we see. We generally see anywhere from 2 to 4 log variation, or in some cases where we don't have a lot of samples, over here farm HA or AD, where we only have 2 samples or 3 samples, it's a very tight group. But in farms that we have a fairly representative number of samples, this is a research site, we have 4 log variation or 2 log variation somewhere in that. And this is then the regional average for actually the Wisconsin set of farms. Drill down now, change to a region in California, but I wanted to also show that their variation, now we already know that there's regional variation, huge amounts, 5 to 6 logs, maybe as high as 7. We actually also see within farm, and now we're actually looking at groups within a farm. And so what this is the number of different California farms that we've sampled. Each of the groups are identified here. This is the terms that they used on farm, and they're representative of at least 10 fecal samples that were collected from each of those different production groups. And you can see, if you can follow my cursor, in this farm called California 3, we see about a 5 log variation within a group of cattle that are in late lactation. Now, some of this is based on feed quality, of course, what they're eating, but it's just an interesting set of data to understand that the prevalence is very high, but the levels within the cows relative to a fecal proxy tell us that the variation between cows is quite dramatic. So what I'm doing now is going to give you an idea about, we talked about the toxigenic side. This is Clostridium perfringens. Rod and Phil outlined that well. And what we did was actually look at Clostridial populations as being 2 types, either on the cow side, over on the left-hand side, we see that 52% of the isolates were found to be Clostridium perfringens, and 47.4% were found to be other than Clostridium perfringens. And we'll kind of drill into what that looked like. On the calves, a little bit more diversity of the others, only 38.9% of the total samples, and the total samples are then shown in N above. You can see we're actually identified as the Clostridium perfringens. When we look at it by region, and here's our map again of the regions, we have the 9 different regions, and these are the actual pie charts from those regions where the total number of isolates, and again, picking 5 isolates from each of the fecal samples. Generally, farms in their formal sampling program get about 10 per production group and about 6 to 7 production groups per farm. You can see it generates a huge number of isolates, and this is the range of data you can see. In all cases, the predominant organism was in fact Clostridium perfringens. That's colored in blue in the code here. And Clostridium perfringens is the 70% in the Western Great Lakes. I think that's the high down to a low of 38% in the I-29 corridor, which is the corridor right here that runs just east to, or north-south, just west of the Twin Cities. So you can see the ranges, and we look at this and to understand that there's quite a bit of variation in terms of what we see by region, generally indicating feedstuff differences, soil differences, again, management that also can play in terms of what impact and what numbers are actually gonna, and what types of bacteria are gonna predominate within that animal system. When we break it down and look at Clostridium perfringens and ask the question, what are the toxigenic types? Within Clostridium perfringens, it's quite boring. 99.2% of the total Clostridium perfringens were in fact just type A. So we, unlike poultry systems and unlike more diverse as even swine systems, cattle systems are very highly dominated by the type A Clostridium perfringens. Now, where do these things come from? Let's look at that from the soil level, and let's look at it in the crops. So this is a chart that was presented in Midwest Animal Sciences this year. We actually show the haylage samples shown here, corn silage in the middle, and TMR. And again, this is a log scale of CFU per gram in terms of total Clostridia population, and numbers in terms of low and high range, and then the end values, you can see it's pretty impressive. So statistically different. Haylage is higher than corn silage, TMR is higher than corn silage, and haylage and TMR are different as well. So what it taught us is that we probably are seeing some outgrowth in TMR. While it makes sense, handling TMR itself, a lot of people think of it as more aerobic, and why would an anaerobic grow? Why would an anaerobic organism grow within a TMR? I think the idea is that what we see in TMR piles is that they oftentimes can generate heat. That heat is actually the end result of bacterial, particularly yeast metabolism. So those yeast are utilizing the oxygen quite readily, as well as the available carbohydrates. They generate that heat and create an anaerobic environment. So it's a perfect environment for Clostridium to begin to proliferate. So that's one of the watch-outs we have for producers when we think about how we're handling that components of the TMR and the TMR itself. Again, a little more detail in terms of the Clostridial loads and feed samples, showing the TMR, corn silage, and haylage. You can see in terms of the range of samples what we actually found. A little hard to see on that prior graph, but this is a nice histogram in the distribution of the actual counts. So in fact, in TMR, 18% of the samples were actually higher than 1,000. And although those numbers seem low, you always got to remember to calculate through on your TMR, for instance. If we would get a TMR of 100 CFU per gram, times the 100 pounds of intake for a high producing dairy cow, converting that from 2 grams per pound so that we understand in the CFU per gram, we get a total CFU now of 4.5 million CFU on a daily basis of a sample that actually has only 100 CFU per gram. So right away, that tells us that one of the challenges we had as really beginning to do research in this area was a lot of microbiologists missed that concept. You know, if you are familiar with silage inoculants and the training that goes on, I think low counts have been misunderstood in terms of their impact on the animal. And clearly through the large amount of intake that occurs daily within high producing dairy cows, we can generate a bacterial challenge of about 4, even with 100 CFU per gram. So it is a significant challenge. So Jesse has presented a number of different presentations at national and regional meetings. This is kind of the summary of those. I think we've hit the high points. We didn't have a lot of time, so we didn't want to spend too much time on that. We do see high prevalence of total Clostridia and Clostridium perfringens in dairy cows. Remember, we were in the high to mid 90s, and Clostridium perfringens is at 80%. A high variation is really the big story here in the total Clostridium as well as the Clostridium perfringens levels. And that variation exists within region, within individual dairy farms, and also within cows within the production groups. So that's really the answer. How and why is that occurring? That's not about just consumption, because even though there may be variation in the TMR, for the most part, when you look at a pen and see that is still 2 to 3 logs, we know that there's some host interactions and other interactions coming into play. And that's really what we need to understand next, is how to control those at the level of the cow. In terms of disease-associated species, so those are the toxigenic. The lead organism was, of course, Clostridium perfringens. It was, in fact, in 52% of the fecal populations of what we surveyed. And again, 99% plus were all type A. And we did see a fairly high amount of genetic diversity within those perfringens. So it's not a single isotype, dominating a system. We see quite a bit of diversity, and that changes over time in general. So what it tells us is, in terms of sensitivity and working at ways to control that, you have to have a product that also then understands that diversity and tries to capture and inhibit that huge genetic diversity of Clostridium perfringens if that's one of your targets. And of course, the next bullet is the other abundant Clostridia detected was the non-toxigenic types, the solvogenic that we've already heard about with Dr. Mackie's speech presentation, the Clostridium baryinkii, biofermentins, which produces 1,3-propanediol, a very potent inhibitor of potentially rumen organisms as well, and then Clostridium butyricum, which is, as in Brod's scenario of good, bad, and ugly, that's on the good side of things. There are differences between regions, as I showed you in those pie charts. There is quite a bit of diversity in terms of the overall, and then also within Clostridium diversity as well. We didn't spend any time talking about beef, but just in case there's somebody in the audience that's interested, we do see that small regional upper Midwest beef feeders are generally like dairies. But when we move to the high plains and look at steam flake corn feeding programs and large feedlot operations, those tend to be even higher in terms of Clostridium perfringens challenge. And in fact, there's a strong correlation to the higher level being in the finishing phase, which in fact is the higher starch. So that sets up the idea about what are the predisposing factors that favor that outgrowth of the Clostridium within the GI tract that will lead to both toxin and metabolite production and potentially impact that host. And as Phil mentioned, it is only probably based on diet, based on host, and a number of other factors that we then can begin to understand what the impact is. But in terms of outgrowth, what we do see is that everybody knows HBS, a slug feeding event due to weather. As high pressure comes in or a weather front comes in, cattle tend to consume. Following, maybe even cold weather in the winter, we see huge off-feed events, and then that feeding event that occurs and gets to a huge slug feeding. Crowding can also cause slug feeding events because animals that are less dominant have to get in and eat quickly. And so those kind of behaviors actually lead to higher challenges for levels of Clostridium. Of course, increased loads of Clostridium in feed is the major driver here. Soil contamination of the feed sources through harvest. We did a nice study with K-State and looked at drive over piles and looked at the ramp, which everybody knows is lower efficiency. We always have trouble when we're feeding out ramps. And is it in fact from the drive over idea that tires carrying dirt and the stickiness of the actual haylage or corn silage pulling dirt onto the ramp and thereby contaminating that ramp with a high level of Clostridium? And we did see some of that in that study as well. And of course, if we're not harvesting at the right moisture, we're not harvesting at the right maturity, and we're not using silage inoculants to drive that pH to help control, we can actually see higher levels of Clostridium in feed. And then one of the other issues to think about is the outgrowth into TMR that I've already talked about. Frequency of defacing fermented feeds is a key thing. When producers are trying to save labor, what they'll often do is say, well, I feed 3 times a day or I feed twice a day, but I'm only going to deface once. Or what we're also seeing on farms is maybe I only want to deface once every other day to save some additional labor. So that that defaced sample sits in a loose pile. We get aerobic organisms utilizing the oxygen, utilizing the available carbohydrates, and then producing a very rich anaerobic environment where we see Clostridial outgrowth occurring. And Jesse and the team has done a number of things there. So that aerobic spoilage actually then begins to favor oxygen utilization and sets up that anaerobic environment to allow Clostridium to grow out. Another one to think about is the bypass starch to lower GI. We've looked at some of the correlations between feed types and different levels, and one of the only things that we can actually find that's correlated to that is actually recalcitrant starch levels. And so there's some good work going on in different diagnostic labs and testing labs that are actually beginning to understand that relationship and begin to understand that starch utilization, reduced starch utilization, could be one of the factors that leads to variations in animals in the same pen having huge differences in Clostridial perfringens and Clostridial total Clostridium in their GI tract. And then one of the other ones to think about is the bypass proteins. Remember Clostridia, Dr. Mackie talked about it in his presentation. A lot of them have the inability to make amino acids, so they don't have the capability of biosynthetically producing amino acids. Therefore, they're scavengers of those amino acids. And if they can find those amino acids, it provides the necessary building blocks for the proteins that are necessary for the rapid growth and proliferation within the GI tract. And of course, we all know stress, pen conditions, stray voltage, etc., increases the susceptibility of the host. We were on a call yesterday with a large group of our large producer in the state of Wisconsin, and they talked about one of the challenges they see anytime they have weather events, and anytime they have some challenges, the susceptibility of the animals actually increases following those challenges to Clostridial type challenges. Not only death loss associated with HBS, but just more digestive issues, off-feed events, and actually could be seen in terms of the measurements in terms of some of the activity meters that they were doing and the health events that they were also measuring. So it's really to think about, it's this cycle that we think about, when we think about where this comes from, we can start on the right-hand side, we can think about manure. So manure is going to contain the Clostridial spores. As we harvest that manure and utilize the nutrients for our crops, we put that back into the field. Of course, the field contains plenty of soil organisms that are also Clostridial, and then we get those into the crops and feed those back. So this is the cycle that we're all trying to deal with in terms of dairy production. And it's really understanding that microbial diversity, that who's there, what are they doing, that's what really we're trying to do. The program that we offer for dairy production today is the Certilis. So this is a bacillus-based probiotic to actually help control Clostridium by producing not only antimicrobials, but helping control some of the leaky gut functions within the cow, as well as some other positive benefits that bacillus can have in terms of the gastrointestinal tract and fibrolytic organisms and a number of other things. So our program is really such that we can start with a dairy assessment, and which is basically we take samples from the farm. We collect the fecal, the TMR, and any forage samples. We analyze those in the lab and report that sample information back. And this is a typical report of what you would see as you get your information from your farm. The Clostridial versus Clostridium perfringens, again, it's the histogram approach. We take a look and then we benchmark it compared to the regional results. So wherever your farm is, we can benchmark it to your region, and we can begin to understand where the risks are in terms of categories, and are they more about total Clostridium or Clostridium perfringens. So another part of the report would include the feed, and in this particular case, we're showing the highlighted yellow because those are the ones that are higher, and just as we calculated in that other slide here, 240 on a high producing dairy cow calculates out to about a 10 million or 11 million CFU total dose on a daily basis for those cows. So certainly a significant part of the challenge. So those are the kind of reports that we do to base the background. What we see in the field in terms of subclinical challenges is much more common than the HBS death loss. And when we feed Certilis, what we actually see is more consistent intakes, decreased off-feed events, decreased GI-related issues, reduced cows in the sick pen, energy-corrected milk response. Generally, in the meta-analysis we've been able to do, we see generally about 2 to 3 pounds of liquid of total milk in terms of energy-corrected. and then more consistent rumination. So that as we get into understanding more of these activity meters and what they can help understand in terms of the impact of Clostridia and solutions to control Clostridia, we'll actually know more, but that's one of the early studies we did, some more consistent rumination and what we see there. And then it reported by producers smoother transitions as well. So with that, I'd like to thank you for the time and your attention, and we're ready for any questions. So I did see some come in. So I need to open up my box. Okay. One of the questions that came in, maybe this is for Rod, but also probably for Phil. What would be the impact of Clostridium population, well, the impact on Clostridium population on available bypass amino acids in the GI tract?
Okay, am I on? Yep.
You're on.
Yeah. So, well, I think it's a good question. It's something that hasn't really been studied, but obviously it could have an impact. So I'd say that generally the Clostridia are well adapted to degrading protein. They're a group that I'd say they're not particularly high in numbers, but they have very high activity, particularly in respect of proteolysis. So I'd say that your higher Clostridium numbers would reduce the post-ruminal flow of protein.
And is it, Rod, that those amino acids would also potentially proliferate or cause the proliferation of the Clostridium populations?
Sure, it's you know, amino acids just don't stick around very long in the rumen.
Yeah, no, I think lower GI tract kind of thing.
Deamination, of which Clostridia are also responsible for that. And so there's correlations. I think we might have seen something in terms of branched chain fatty acids, deamination of the branched chain amino acids. But I think those are things that really need more study. So it's potentially possible, yes.
And Phil, anything you'd like to add there?
Sure. Yeah, I think for example, in our model where we are feeding a good amount of rumen degradable protein, we're feeding rumen protected amino acids, and I don't think we're giving, and we are talking about mid to late lactation cows. You're not short of those amino acids, so I think that would be more important transition and maybe fresh cows or cows at peak that you'd see perhaps some of those indications on milk protein or casein in relationship to milk protein that could, in a case that you're challenging with some of those Clostridium, that you could see that. But yeah, I think you'd have to be a unique situation for be so aggravating that those bugs would make the cow suffer for not having those amino acids.
But there may be some relation to ammonia levels and the milk urea nitrogen.
Okay.
That you pointed out.
Which we did see in the study. Phil, this is for you. The lactation trial, did we collect milk components, and what were the differences, if there were any?
So the only tendency that we had was for milk urea nitrogen.
Okay.
For the cows challenged having a little bit less of that milk urea nitrogen than the control. So we had protein, casein, fat. That was all. There was no difference.
Okay, very good. And Phil, in terms of your findings, both on toll-like receptor 2 and then also your proposed idea about shifting the metabolism probably to a more propionate type, as you see that variation day to day, what you were saying, well, I can't definitively say that they're detrimental, but I think what we're seeing is that it might be transient in terms of impact. One of the things we haven't shared with this group is the results of the effect on the fibrolytic bacteria. Although we didn't see differences in this particular trial, we have seen trial results where we actually feed Certilis and actually see an improvement. We've always had the hypothesis that the Clostridia populations would have been impacting that possibly through solvogenesis and other metabolites that impact those. Any thoughts on, you were kind of wrapping around the idea that perhaps there'd be higher fiber utilization in some of the cows that are actually treated with in the experiment where we challenged the cows with the Clostridia.
Yeah, and I think that that's what I'm thinking. So there's definitely something there when you find those guys and when you find the variation, but I'm not sure if they are directly causing the problem. And I think that's what we're talking about, or they are an indication that something else is shifting. And that's where we are seeing the problem. But I think that's when we talk about starch and we talk about the fiber, there's a component that we don't know a lot about. That's the sugar component, and sometimes that's just called soluble fiber, or is thrown on the same thing. So when you were talking about the starch on your last slide, that got me thinking that you may have some sugar components there, components of the fiber that may be involved, or this non-toxigenic somehow, Clostridium, somehow is directly using that, or is impacting somebody in the rumen that now it's able to be more consistent or something like that. And I found interesting, and again, I don't know anything about that other trial that I showed from Argentina, even though I think the product is made here in the US, but in the addition with yeast, right, that is supposed to help even more in breaking down some of those fiber components. I think there's something to it as well. But yeah, so I think that whole relationship of the TLR it's changing, right? So it seems like you're having less of that communication needed, at least when you feed this non-toxigenic. Does that mean that it's passing through the rumen in a different way, in a different pace, or is just a different composition, and then you're getting all the effects happening at the intestine? But the rumen papilla is changing, right? And that association of, hey, you had this higher tight junctions, and that's associated with more acidosis. So all matches for an environment that is changing to be more prone to starch or sugar digestion after the fiber was done. So, and I think that's mainly is happening in the rumen, right? Because you don't expect that to be happening down there in the GI tract.
Mm-hmm.
So do you see, for example, Tom, I don't want to put you in the spot here, but let's say if you have, because the other thing is, we actually, when we challenge those cows, we are having like a pure 5 strain or a few strains of that particular Clostridium that usually that maybe not happening at the commercial farm, right? At the commercial farm, they have a whole population, a whole juice of other things. They don't have that thing specific. If you had that Clostridium bifermentis and Barankia sporulate and make it like a DFM, what do you think would happen if you had that as a top dress and gave that to transition cows?
Yeah, our understanding is that in general, the non-toxigenic, that's the big question. We that's what we're trying to answer by getting this particular study done with you guys. So I don't know. Right now, there's some deleterious things that you're seeing relative to the toll-like receptor, relative to some of the expression of certain key genes on the papillae surface. I think those aren't positive, but yet it would be interesting to see in different stages of production where the impact might be and what that would be. So, as you said, we really looked at cows that weren't in transition. We also looked at multiparous cows. So, I mean, what's primiparous look like? What does a transition cow look like? Those are all questions. When we, look on commercial level and think about where we see clostridial challenges, and farms report back to us in terms of, especially if it's a non-HBS farm, it's a farm that has metabolic problems, digestives, a lot of pulls, that's what they describe. And generally how they describe their problem, we can predict what likely the outcome will be in terms of Clostridium perfringens versus the non-toxigenic types. And it's very rarely that we miss that. So when we hear digestive and no death, and we hear off-feed events and other little problems, we usually think, okay, they're really not fighting Clostridium perfringens. Their dominant organism might be, in fact, the non-toxigenic. So those are easier farms in terms of overall problems because they're not as severe. Whereas farms who report, a death loss of maybe even as high as 3 to 5% on a monthly basis, we know we're walking into a Clostridium perfringens farm. And it tends to be that you have farms that are either or, and then there is a little bit of it's kind of 50-50, but it generally is, a farm is generally Clostridium perfringens challenged or the non-toxigenic challenged, and the middle ground is not as frequent as the two extremes. But in the end, to answer your question, the answer is we don't know. And that's really what we're trying to do with this kind of research, is begin to understand what the impact of those Clostridium are. And it is very complex. And by us taking 3 strains of multi-species and saying, okay, we can hope that predicts what's going to happen in nature, I think we're kind of approaching it from a pure culture microbiology standpoint in a very diverse and complex world of multiple strains, multiple species, and likely polymicrobial challenges to boot. It's not just about that. There's other things that dairy cow is dealing with, various levels of enterotoxigenic E. coli, as we see in most dairies, fairly high. So all of those things interact and predict. And I think that's why when we start to look at pen-to-pen variation being 2 or 3 logs in terms of Clostridial loads in the fecal samples, that tells me that some hosts are fighting that off well, and other hosts are probably succumbing to those populations. And there's a great article reference that we can give the attendees from Goosen. It's a group out of Belgium. They've got a great visual of a series of allele loop experiments in which they actually inoculate those with 1 million CFU and watch it over time. And even in 30 minutes, the epithelial surfaces from those villi and microvilli start to slough off based on Clostridium perfringens. So perfringens is the big damaging causing. The others are not, but they have an effect, and what that is really what we're trying to figure out next. So with that, I appreciate everybody's time and efforts, and thank you all for attending. And you'll see an invite soon on the 5th of the Arm & Hammer seminar series coming soon. So thank you for your time, and have a safe and good afternoon.
Okay, thanks everyone.
Thank you.